This is Wendy. Wendy went on a holiday with her two daughters that you can see there. She contracted a bacterial infection, went into hospital, and was given vancomycin. The vancomycin, unfortunately, was a toxic dose, and she died. That death was preventable. This is Jim, not his real photo. Jim was at work and had chest pain. They sent him to hospital, where he waited 4 to 5 hours to have 2 blood draws to confirm his heart attack diagnosis. In that period of time, his condition deteriorated and he died. Jim's death was also preventable. At Nutromics, we exist to save the lives of people like Wendy and Jim. And we have a vision to create a world with 0 preventable deaths through continuous diagnostic monitoring. We started the company, Hitesh in the audience here, myself in 2017 with this very vision. And we set about from that moment to find a real-time continuous diagnostic monitoring technology that could fulfill that vision. 2 things that we were looking for. Number 1 was it needed to be demonstrated to work in vivo, and we found several technologies that could do that. And number 2 is we needed to be a platform that could monitor any molecule. We spent a lot of time looking at a lot of different technologies, and in 2020, February, right before the pandemic, we secured one technology, DNA sensing, that could do both. It is both demonstrated to work in vivo and a platform technology that can monitor any molecule. So since then, we've built a company and a team of 60, and we've developed a medical device that I have in my hand here, and it's comprised of microneedles, and on the tips of those microneedles, we put our DNA-based sensor. And it's what we call, in effect, a lab on a patch. It has 4 major attributes. The first one is multi-analyte. So we can monitor drugs and biomarkers. The second one is multiplexing, meaning on this one device, we could put multiple sensors. Thirdly, we do all of that continuously and in real time. And lastly, it's autonomous, meaning it's set and forget. Clinician puts it on and they walk away. So how does this all work? This 1-minute animation puts it all together. So we're in the ICU, which is where we're going to start. Patients getting dosed a drug that requires therapeutic drug monitoring. And we're going to go into the interstitial fluid, which is where we operate. These are the microneedles, and on the tips of those microneedles is the DNA-based sensor. Very selective to its target. So you can see other targets come along and doesn't respond. Target of interest now comes along, vancomycin in this case. It recognizes that. There's a signal change, which we can monitor. It releases it, which is what allows it to be continuous. Each one of those needles is a separate sensor. So we're the world's only capability for multi-analyte sensing. Signal goes through a Bluetooth gateway out into the cloud and back down into pre-existing screens or the handheld hub that we've created. So in a very simplistic way, that's how it's all been put together. Now that's a really nice animation. Does it work in real life? So the answer to that question is a resounding yes. In the last 2 years, we've been in clinic and we've been running clinical studies on the device. And what you're seeing here is the world's first monitoring of a drug in real time. So that's a game changer when it comes to drug dosing, the mainstay of hospital. And the data is very high quality. You can see there the data from one of those individuals in the study. So first question, do we have a technology proven to work in vivo? Check. Second question is, do we have a platform that can monitor many different targets? Well, together with our partners, we've developed dozens of these sensors and validated 24 in vivo. So the answer to that question is yes. So if you have a platform technology that can monitor any drug or diagnostic target, what problems are you going to solve in healthcare? That's a question we thought long and hard about. And where we're starting is therapeutic drug monitoring of the antibiotic vancomycin. That's the drug that killed Wendy. It's commonly dosed. 1 in 6 people get this drug. It's used for life-threatening bacterial infections like sepsis and MRSA, and it's exceptionally difficult to dose this drug. How is it dosed? So you take a blood draw, someone carries that sample to the lab where it's processed, and then hours later, the doctor gets a single data point for vancomycin of where that patient was when the blood draw took place several hours ago. So what the doctors are trying to do is get these patients into this green, narrow therapeutic window where it's killing the bacterial infection. And what they see are these little red dots. That's where the blood draws are taking place. But they're seeing that in the rearview mirror. What they don't see is what's happening in between, all the variability in that drug concentration. So the outcomes are very poor. Up to 43% of patients get a vancomycin-induced acute kidney injury in the ICU. A third of them die, and each one of those acute kidney injuries costs, on average, $14,000. So what do doctors do? They're traditionally conservative. They don't want to give a toxic dose. And that has another problem with underdosing, because you're trying to treat a bacterial infection that's life-threatening. So what we're going to do is we're going to solve that problem with continuous real-time monitoring of vancomycin on this device. And we're going to take something that looks like this and turn it into something that looks like this. Far greater control in the therapeutic zone. The benefits for the patient are obvious. Significant reduction in acute kidney injury. The benefits for the doctor are a simplified workflow. In fact, it takes less than half the time to dose vancomycin with this device than current standard of care. And for the hospital, we're reducing one of a very expensive acute kidney injury. So the return on investment is very strong. So we're just talking about vancomycin here, but we have a platform. We can go beyond that. We have 13 drugs queued up like vancomycin that have narrow therapeutic windows and high toxicity. We can also do biomarkers like creatinine. So how are we going to roll this out? We're going to start with infectious disease vertical, then move to cardiac, and then renal, and solve some of the biggest challenges we have in hospitals. We calculate there is a circa $100 billion TAM opportunity in doing so. We're going to manufacture this device ourselves. So this is a few of the team at our equipment manufacturer in Europe. And we build this machine once and we leverage it over and over again because it can do multiple product variants. Each one of those, by the way, COGS, we're driving to sub $10 with a gross margin above 80%, so very healthy unit economics. We're the world leader in DNA sensing technology. As I mentioned before, we secured the tech in 2020, and we've now got 32 patents enveloping that. We are the first and only company that's demonstrated this technology in humans, and we've raised 5 times more capital than our nearest competitor. So we're proud to announce today that as of a few days ago, we just secured the lead investor on our recent acceleration round, which is circa $20 million. In a couple of weeks, we're moving into the ICU, our intended use population. And off the back of that value inflection, we'll be opening up our B round later this year or early next year. We'll have early revenue in 26 and full commercial launch in 28. Today, there are millions of Wendy's and gyms that are walking around that are relying on this product to survive. And together, we invite you to join us to create a world with 0 preventable deaths through real-time diagnostics. Thank you.